Hello, everybody. My name is Marcia Higas, and it's my absolute honor to introduce today's speaker, Dr. Celeste Simon. Celeste received her PhD from Rockefeller University and performed her postdoctoral work in the lab of Dr. Joseph Nevins. She then worked as a Howard Hughes associate in the lab of Stuart Orkin. She started her lab at the University of Chicago and in 1999 moved to the University of Pennsylvania, where she was an investigator of the Howard Hughes Medical Institute for 20 years. She's currently the scientific director of the Abramson Family Cancer Research Institute. It's really a thrill to introduce Celeste. She's been a personal scientific hero, and I'm really honored to consider her a friend and a colleague. Her work has been absolutely seminal in uncovering the role of, helping to uncover the role of hypoxia signaling in physiology and cancer. And she has just, always throughout the years been at the leading edge and the frontier of really exciting biology and physiology, including new metabolic studies. So Celeste has received a number of awards, and it's too numerous to go through. I'll just mention a few notable awards. One university named her Woman of the Year. That jumped out to me. She was recently elected to the National Academy of Medicine. She was funded as a Howard Hughes Medical Institute investigator for 20 years or so. She recently was awarded an R35. And so I'm so excited that you're speaking today, Celeste. Thank you so much. And thank you all in the audience for participating in today's seminar. And I want to mention first, as you have questions, please submit your questions via the Q&A tab. And after Celeste's seminar, I will go through and help moderate the question session. Thank you.
Thanks a lot. And I am always delighted to be in Boston, either really or figuratively. And I do have a brief infomercial. If anybody in the audience heard a presentation recently in Dr. Kidakowski's PO1 meetings on Wednesday, I would suggest you actually go get lunch now, because this is going to cover some of the same material. But having said that, the title of my presentation is Hypoxia, Metabolism, and Tumor Progression. And as you can see, I've highlighted the word hypoxia, and I did this for two reasons. The first reason is I'd like to describe how our own longstanding interest in studying this topic has led to the current area of research, which is metabolism and tumor progression. And secondly, I'm still celebrating the 2019 Nobel Prize in Physiology or Medicine awarded to 3 hypoxia researchers, including your very own Bill Kalin. So it now seems like a century ago, but it was still very exciting for us in the field. And at this time, anything that lifts spirits is really appreciated. So we've been interested in tumor progression because, let's see now, I'm not getting my slides to advance. Here we go. Okay. Because of the following features that are exhibited by most solid tumors. So for a number of reasons, including deregulated rates of cancer cell proliferation, the altered metabolism of both tumor cells and the recruited stromal components, and these would be endothelial cells, immune cells, and reactive fibroblasts, and what is typically gross vascular insufficiency. Most solid tumors, as shown here for a growing metastatic renal cancer, develop domains that are severely limited for oxygen. That is, they are quite hypoxic. And so for the disease to progress, the malignant cells have to very carefully balance the oncogenic demand to expand biomass and undergo cell division, but somehow still remain viable in what's intrinsically a harsh microenvironment, where the cells are not only hypoxic, but they're also starved for nutrients. Now, recent work from labs such as Matt Vanderheiden, by measuring interstitial fluid of, for example, pancreatic cancers versus healthy pancreata, indicate that what the nutrient scarcities are may need to be revisited. But nevertheless, I think invariably solid tumors are going to have domains where cells are very limited for oxygen, and some combination of scarcity for glucose, glutamine, other amino acids, and lipids. And this is something that the malignant cells have to somehow overcome and continue growing. And then finally, of course, the malignant cells have to evade the immune system. And so today we're going to discuss how metabolic transitions and stress responses contribute to all three things. So what might these be, again, for a growing metastatic renal cancer? And we've argued for some time, and I think we've been able to demonstrate that virtually all of the homeostatic pathways are affected by nutrient and or oxygen deprivation. For example, the central nutrient sensor, mTOR, is inhibited by hypoxia in a matter of minutes, even if the cells have ample access to glucose, amino acids, and insulin. Hypoxia promotes an unfolded protein response because oxygen and glucose are needed for protein maturation. It's clear that autophagy is stimulated. And this very interesting cell biological process known as macropinocytosis, where cells simply engulf nutrients from their surrounding interstitium, is clearly enhanced by nutrient scarcity, but there's increasing evidence that even oxygen contributes to this very interesting process, a typical feature of, for example, RAS transformed cells. Now, there are at least 70 O2-dependent dioxygenases, and recent work from Bill and Sonia Rocha, and now some others, has shown that at least some of these, particularly those that regulate the status of histone methylation, have low affinity, i.e. high Km for molecular oxygen, once again indicating that hypoxic environments will contribute to the epigenetic status of cells and maybe influence cancer epigenomes. And unsurprisingly, a number of metabolic pathways are either directly or indirectly influenced by low O2. And something we published some time ago is a good example of that. And this is a simple biochemical reaction. It's how an abundant saturated fatty acid, like stearate, is converted to a monounsaturated fatty acid, like oleate. In fact, oleate is one of the most abundant of these kinds of long-chain fatty acids. And this enzymatic reaction is regulated by an enzyme called steryl-CoA desaturase 1, or SCD1. It's an oxygen-consuming reaction. And we've been able to show that oxygen availability is limited enough in a number of solid tumors like renal cancer in such a way that lipid desaturation is inhibited. This leads to an unhealthy buildup of saturated fatty acids versus unsaturated fatty acids, and changes in membrane integrity, changes in the endoplasmic reticulum, and something we call lipotoxicity, which is something we're continuing to study in a number of cancers. Now, of course, one of the best characterized responses, I think, needs no introduction to this audience. This is the large transcriptome regulated by hypoxia-inducible factors, or HIFs. And I know everybody's very familiar with this, but just very briefly, the HIFs are heterodimers consisting of a constitutive subunit we call either HIF1 beta or ARNT, or the HIF alpha subunit, which can be either HIF1 alpha or HIF2 alpha. And the pathway is stimulated in a very simple way, such as when oxygen is plentiful, this alpha subunit is very labile, but when cells become hypoxic, or in the setting of a heritable cancer syndrome called von Hippel-Lindau syndrome, where the E3 ubiquitin ligase recognition component, VHL, a tumor suppressor, is lost, this leads to the reversible stabilization of HIF, its dimerization with ARNT in the nucleus, and increased expression of thousands of target genes. So this is a very essential transcriptional response that promotes hypoxic adaptations. And among these targets are metabolic enzymes, which we'll discuss a little bit later. But the highly conserved and important pathway that includes HIF, the prolyl hydroxylases, and VHL was recognized in 2016 by the Lasker Committee. And this was awarded to not only Bill, but two other friends, Peter Ratcliffe and Greg Semenza. And I wrote a short piece on this in the New England Journal of Medicine, and they hired a graphic illustrator that put together this really quite beautiful diagram, even though it's a little bit busy. But then I'll contrast that with the artist that was hired by the Nobel Committee in 2019. And you guys know Bill, he's much more handsome than this, as are Peter and Greg. But never mind, I think this was just a wonderful day, not just for them, but for all of us in the hypoxia community. And I even benefited more because the committee invited me to attend the ceremony in December in Stockholm. So this is the ceremony at Stockholm Concert Hall. In fact, Bill's sitting right in there. You're not supposed to take pictures, but I snuck one. And then this is actually at the Nobel banquet at Stockholm City Hall, and I'm seated next to Randy Johnson, another good friend in the hypoxia community, who actually announced that Bill, Peter, and Greg had won the award. And I will say that it was only last December, but in the current pandemic, it really does seem like a galaxy far, far away and a long time ago. But anyway, coming back to stressful tumor microenvironments. So I think this audience would appreciate that solid tumors are not only genetically diverse, they exhibit tremendous intratumoral heterogeneity. And the heterogeneity at the genetic and epigenetic levels is further compounded by the heterogeneity of different microenvironments in tumors that have variable levels of oxygen and nutrients. Even with that in mind, we have set out to ask this question that I think we can make a good argument for, and that is, do disparate oncogenic changes and heterogeneities in these tumor microenvironments nevertheless converge on more common metabolic adaptations, and are these actionable? And so that's what I'd like to discuss today. And one of the platforms we've used for this is in the setting of clear cell renal cell carcinoma for the following reasons. So again, I think because of obesity and longer lifespans, this is a tumor whose incidence is on the rise for both men and women. It has a 5-year survival rate of about 85% if highly localized, but as is always the case, survival rates plummet in the setting of metastatic disease. Outside of gemcitabine, it's thought to be resistant to both chemo and many forms of radiotherapy, although I think you can appreciate that immunotherapy is really showing great promise. And in fact, immunotherapy combined with angiotherapy is looking very promising. Nevertheless, we still have patients that either stop responding or don't respond, so there's still work to be done. Now, we were drawn to this disease some time ago for the following reasons. These tumors tend to lose VHL function in better than 90% of the cases, suggesting that they would have essentially constitutive HIF activity, although I'll modify that in a moment. And we already knew that they had a very consistent metabolic change. That is, they tend to accumulate vast quantities of neutral lipids, including triglycerides, cholesterol, and cholesterol esters. And in fact, we showed some time ago that that made these cells strictly dependent on perilipins, which promote lipid droplet formation, which sequester these neutral lipids away from the rest of the cytoplasm to, again, avoid lipotoxicity. So with this in mind, we and others have actually contributed to the, my phone was ringing. Anyway, we've contributed to a lot of omics characterization of these tumors. We have transcriptomics, we have copy number variation data, we have exome sequencing data, And we embarked on a pan-metabolomic assessment, first with 20 matched pairs, and then this was repeated with roughly 140 matched pairs, simply comparing the metabolomes of healthy kidney tissue versus tumor tissue. And it's a snapshot, but we're simply looking for consistent changes. And of course, one consistent change is a buildup in the tumors of metabolites that fall in the categories of glycolysis, gluconeogenesis, and glucose-related sugars. Now, we integrated that with a metabolic gene set analysis of now about 500 independent cases and roughly 80 healthy tissues. And what you can see, many of these metabolic pathways in the tumors are in fact underexpressed. Now, this seemed counterintuitive because I think we would imagine that solid tumors have a revved up metabolism. In fact, this comes back to my first point. We think malignant cells reside in very stressful microenvironments, and so they have to carefully modulate their metabolic activity with what the environment can support. The most underexpressed categories shown here are a series of enzymes involved in what's known as gluconeogenesis or carbohydrate storage. And one of them is the enzyme known as fructose 1,6-bisphosphatase or FBP1. Well, what can I tell you about these enzymes? So, coming back to typical features of cancer metabolism, I think you're all familiar with the notion of Warburg metabolism. That is that solid tumor cells or many malignant cells of any kind tend to be quite glycolytic for a lot of reasons, including the production of building blocks to make nucleotides, fatty acids, and amino acids. Now, Ralph de Berardinis, working with human patients and metabolic labeling on primary patient materials, has shown that renal cancer actually does fit the classic idea of Warburg. They're very glycolytic. They catabolize glucose, but they tend not to oxidize it. So if these tumors are glycolytic, it would certainly stand to reason that the enzymes that do the opposite, they don't catabolize glucose, they generate glucose from non-carbohydrate substrates. It would certainly stand to reason that all three of these enzymes shown in green would be downregulated. So nothing surprising there, but it turns out FBP1 is a special case. And that is for the following reason. So if we simply re-express FBP1 in renal cancer cells that don't have it, we can show that, as we anticipated, cell growth is slowed a great deal. But the surprise came when we re-expressed a catalytic activity dead version of this protein. And even though they suppressed cell growth, the catalytic activity dead version was not as potent. And we then subsequently learned, much to our surprise, and there's a growing number of metabolic enzymes that have these properties, that FBP1 is not only the cytoplasm, it's actually in the nucleus. Even more amazing to us, it's physically associated with chromatin. And finally, it's directly binding HIF1 and HIF2. And we've been able to show this by either co-IPs or GST pull down of bacterial expressed proteins. We've been able to chip FBP1 and HREs and chromatin. We can re-chip FBP with HIF-SYK chromatin. So there really is a direct physical association, which would further suggest why FBP1 expression is so systematically suppressed. You're reinforcing glycolysis, and you're releasing their interaction with the HIFs, because if they bind to the HIFs, they actually attenuate their activity. So I'd like to tell you a little bit more about what we know about the FBP-HIF interaction. As we studied FBP and its relative FBP2 in more detail, we realized that they have quite well conserved nuclear localization sequences. These can be mutated, and this keeps the FBPs out of the nucleus if they're expressed. Well, FBP functions largely as a tetramer, and it can be subdivided into an N-terminal regulatory domain and a C-terminal catalytic activity domain. And I'll simply note that there's the allosteric inhibitor, AMP, that binds in the N-terminal domain here. Now, what's shown in this part of the slide, quite simply, is the catalytic activity dead version of FBP, And this shows that indeed it doesn't retain any residual enzymatic properties. Using a very simple HRE luciferase assay, I can show you that when we re-express FBP1, remember the renal cancers have lost it, so if we re-express it in these cells, you can see that you don't completely shut off HIF activity, you simply attenuate it about 40%. The catalytic activity dead version of FBP is essentially just as good. So this is a catalytic activity independent property of FBP. It can go to the nucleus, it can associate with HIFs at HREs, at chromatin, and simply dampen their output, and this is completely independent of their enzymatic properties. So where are HIF and FBP interacting? And so I'm now showing the topology of FBP as shown here, and we simply made systematic deletions in the protein. And going back to our HRE luciferase assay, it was very clear that you lost the ability of FBP to attenuate HIF output if you deleted either exon 1 or exon 2. So this is now showing how well conserved FBP1 is, which is primarily found in kidney, liver, and intestine, versus FBP2, a more generally expressed fructose 1,6-phosphatase, which is found in a number of tissues, especially muscle. And they're highly conserved. However, they do share some leucine residues, again, more in the N-terminus than anywhere else. And we further queried the properties of FBP1 in its ability to physically bind HIF. And here I'm simply showing bacterial expressed proteins to make the point that this is a direct physical interaction. And so if you take FBP1 and show that, basically make these mutations in a series of amino acids, in particular, these two highly conserved adjacent lysine residues, the ability to associate with HIF is significantly lost. And I'm simply highlighting that here. So either lysine 72 or lysine 73, the changes don't really affect FBP1 HIF interactions as indicated here, but if you mutate both of them, their ability to associate is significantly lost. And now if we go back to that simple HIF luciferase activity assay, so here's HIF activity in control cells, its attenuation with wild type FBP1. The attenuation is somewhat restored if we introduce an FBP1 with those two lysine residues mutated. Now this mutant, I'll come back in just a moment. So coming back to the topology of FBP1, as I mentioned, AMP, the inhibitor of FBP, and if we just take a step back and think about this just a moment, I think that you can appreciate that it would make sense that AMP inhibits FBP1 activity. So if cells are in a good bioenergetic state, it would be fine to generate glucose because they could handle that in a bioenergetic situation. But if they become bioenergetically challenged, then you certainly don't want to make glucose, you want to catabolize it. So if AMP builds up, it is quite logical that the activity of FBP1 would be inhibited. But does AMP actually disrupt the FBP1 interaction as a way to sense the metabolic status of these cells, that is, the status of low energy? And we noted that where HIF is binding to FBP is roughly in the vicinity of where the AMP interaction is thought to take place. So to look at this in a little bit more detail, we went back to those in vitro pull downs, and while these assays are very difficult to do, we can simply dial up the amount of AMP, and as shown here, the ability of HIF and FBP1 to associate is attenuated. Well, what about where AMP actually binds to FBP? So again, another series of point mutants in FBP to show where AMP association takes place. boiled down to this threonine residue 27 as being critically important. So finally, when we go back and introduce this mutation into the wild type FBP1 protein, as indicated here, you can see that the wild type FBP1, for example, or other mutants, if AMP is added, the ability of FBP1 and HIF to associate is somewhat attenuated. Again, these experiments need to be repeated in live cells, I think, to make them even more impressive. But what's clearly shown here is that the point mutant that causes FBP1 to no longer bind to AMP now loses that effective AMP on FBP1 HIF1 alpha interaction. And then further shown here, again, wild type FBP1 is attenuating HIF activity. The mutant of FBP1 that no longer binds AMP, if anything, is even better able to attenuate HIF. But the protein that now can no longer bind HIF1 has lost its ability to attenuate HIF output. So to try to just simply put this all together for you, we think that in a high energy state, if the HIF alpha ART dimers are present and binding HREs, and this would be particularly the case in cells that have some constitutive HIF activity, FBP1 is associated with the dimer, somewhat attenuating its transcriptional output. But then if the cells experience some kind of bioenergetic challenge where they have low energy, where AMP levels are coming up, this would then dissociate FBP1 from chromatin, and now we really do have full-on HIF activity. And now if we simply circle back to renal cancer, it certainly makes sense why these tumors have very consistently lost FBP1. We've looked at over 1,000 individual patient samples. And that is the epithelial cells of the proximal tubules, the presumptive cell of origin for renal cancer, have FBP1 in both the cytosol and the nucleus. And in the cytosol, this allows a very careful balancing of glycolysis versus gluconeogenesis. And we're now further probing other nuclear activities for FBP1, but the initiating event for this disease is to lose VHL, and we've now shown that they must lose FBP as the next step, because now you really do have full-on HIF activity, you reinforce glycolysis, and the disease progresses from there. So FBP1 is consistently lost, either by copy number loss or epigenetic silencing, because if it were present, it binds at HIFs at their HREs and chromatin, attenuating their activity. So then we made a conditional allele of FBP1 because we've all been very keen to finally have a workable genetically engineered mouse model of renal cancer. This has remained elusive in a tractable way, and I'd be happy to chat about that in the Q&A if people are interested, but I'd like to go to a few other points and then begin to discuss some brand new data. So while we were trying to get our FBP1 allele into what we thought were the beginnings of clear cell renal cell carcinoma, genetically engineered mouse models, we also decided to look at another cancer, which is liver cancer. Now, liver cancer is the most predominant form, hepatocellular carcinoma is the most predominant form of liver cancer. Its incidence is also on the rise due to many factors, including obesity, diabetes, and alcoholism. There are treatments to offer patients, but unlike renal cancer, which still has quite a few things that the patients can contemplate, once HCC progresses beyond phase 1 or stage 1, it's pretty hard to treat. So the medical need here is really quite substantial. Immunotherapy is beginning to show some signal, but my read of the clinical data is we have a lot of room for improvement. So with liver cancer, we did the same thing. We looked at a large number of transcriptomes comparing HCCs to healthy liver, and the same trick. This pathway was again very substantially downregulated, but the opportunity for us is that unlike renal cancer, HCC has a number of very well validated genetically engineered mouse models, And this allows us to begin to look at some of the things that we're very interested in doing, i.e., what is the metabolic exchange between the tumor cells themselves, in this case, the HCC cells, and the other constituents of the microenvironment, the recruited immune cells, for example, the endothelial cells. And so we can take our FBP1 conditional allele and cross it into a number of genetically engineered mouse models, or those that are simply treated with either the carcinogen diethylnitrosamine or exposed to liver damage and high-fat diet. There's a long list of HCC models, in particular those that are good mimics of what's known as non-alcoholic fatty liver disease, a clear metabolic predisposer to HCC. We've recently published this work, so I'll just summarize it somewhat briefly, but I think this slide makes the very clear point that if you lose FBP1 in these models, it greatly accelerates the disease and significantly enhances disease burden. We showed that in models that included those that were more diet-driven. So this is a model where the mice are exposed to high-fat diets of various shapes and sizes, and again, disease burden is increased. So what we've just recently published is that in liver cancer, FBP1 expression is also very consistently lost. So this is another very consistent metabolic adaptation, just as we noted in kidney cancer. But in our mouse model, if we simply delete FBP1 to begin with, it further enhances disease progression. So rather than having the slow epigenetic silencing of the locus, it's gone. And so this greatly promotes liver tumorigenesis. Now, the next thing we really wanted to do is do immunophenotyping and see what in the world happened to the immune response to HCC. And I'll talk about this a little bit more, but we didn't see big differences in numbers of a number of these different immune populations. Doesn't say anything about functionality, but there were clear differences in an NK cell population and myeloid-derived suppressor cells, and I'll talk about that in a moment. The low-hanging fruit, however, was another constituent of the tumor microenvironment, and that is the so-called hepatic stellate cells. So if you're not familiar with them, these make up about 10% of liver mass. In a healthy liver, they tend to reside between the hepatocytes and the endothelial cells of the liver sinusoids, and they're in a quiescent state until there's some kind of injury, which could include, of course, oncogenic stress. And so these cells become activated, they become myofibroblasts and contribute to fibrosis, which can also greatly contribute to cirrhosis, another metabolic predisposer of liver cancer. Well, we performed a secretome of FBP1 hepatocytes, primary hepatocytes, compared to controls. And we noted that included in the secreted material are factors such as high mobility group protein 1 or CCN1, and that these actually seem to be responsible for activating the stellate cells and then causing them to senesce. And indeed, the FBP1 loss in the hepatocytes encourages the stellate cells to activate, then senesce, and then make a secretory phenotype known as the senescence-associated secretory phenotype. And many of these factors are known to promote liver inflammation and greatly enhance the disease burden of hepatocellular carcinoma. Furthermore, the transcriptome, very interestingly, showed enhanced expression of lipogenic genes. So simply deleting FBP1 led to liver steatosis, a buildup of neutral lipids in the hepatocytes. This brings up another very interesting point, and that is that we believe that loss of FBP1 in the hepatocytes is not simply allowing glucose-derived carbons to get repurposed for lipid synthesis. We think that in the liver, once again, FBP1 is in the nucleus, talking to nuclear factors. And this allows me to just briefly mention work on the related factor that we've just published. This is the protein FBP2. So FBP2 is highly expressed in muscle and myogenic progenitors, and in sarcoma, a tumor of this organic site, FBP2 is also very consistently lost. And we found out that if it were present, it would associate with chromatin, but not with the HIFs, in this case, actually with c-MYC. And so if it associates with c-MYC, we can show this by CHIP, by CHIP-RECHIP. The point being that this EBOX in a promoter of TFAM, which I'll come back to in just a moment, not only binds MYC as we predicted, but it also binds FBP2. And if FB2 is bound, it attenuates c-MYC activity. So what does this mean for sarcomagenesis? And I'll just simply summarize here because I don't want to go over my allotted time and wanted to get to a final story. So as I mentioned, in renal cancer, if FBP1 were present, it would bind HIFs and attenuate their activity. Well, similarly, in sarcoma, if FBP2 were present, it would bind to MYC and appears attenuate its activity. And so the reason why FBP2 is so consistently lost in sarcoma, like FBP1 is lost in both renal cancer and liver cancer, is that you not only once again promote glycolysis at the expense of gluconeogenesis, but now you release any attenuation of c-MYC upstream of factors like TFAM that are responsible for mitochondrial production, mitochondrial biogenesis, and mitochondrial function. So we suggest, and our ongoing experiments in liver cancer lead us to believe that the nuclear FBP1 in liver cancer, its loss is now revving up MYC activity, which is contributing to lipogenesis. And in the FBP1 deleted hepatocytes, we see a significant buildup of a number of lipids, again, a predisposing metabolic syndrome for hepatocellular carcinoma. Now, I just want to take a step back. You know, we're working with two very abundant metabolic enzymes. Indeed, they have nuclear localization sequences. We're doing a lot of work to fully understand their nuclear shuttling, but you might be skeptical that these two metabolic enzymes we stumbled on through unbiased approaches are talking to two of our favorite proteins, HIF and MYC. So we want to do due diligence to make sure that all of this is correct and we're not just looking at sticky proteins, but I think our work is consistent, and the onus is on us to really further prove these points. Now I have about 10 minutes left. I'll just mention that the senescence of the stellate cells suggests a therapeutic opportunity, and that is so-called senolytics. So we've done this two ways. We can treat our mice with the combination of dasatinib and quercetin and greatly attenuate disease. We've also done this with a more standard senolytic called navitoclax. And so the conclusion for these studies is, as I said, FBP1 loss promotes HCC progression. It results in a big change in lipid metabolism, including ceramides, which we think actually cause further lipid damage, liver cell damage. This releases factors that engage the stellate cells, causing them to senesce. And we're seeing effects on NK cells, which usually respond to the senescent stellate cells to reverse liver injury, but we think the metabolic changes here are subverting their activity, and we're currently looking at that. And I'll also note that the senolytic treatment, which shrinks the tumors, coincides with a big influx of T cells. So one possibility might be, could the senolytics perhaps be combined with immunotherapies and improve their effectiveness in this disease? In the few remaining minutes, I'd just like to come back full circle to another consistent metabolic adaptation that we're seeing in renal cancer. And as I said, this has been appreciated for decades and decades. They build up cholesterol and cholesterol esters. And the assumption was that this would help cell proliferation. It'd be a great fuel for the mitochondrion. But again, it's very clear that the mitochondria of renal cancer are dysfunctional. They tend not to engage in fatty acid oxidation. Nevertheless, there's this very consistent import of cholesterol esters, and their accumulation to the point that it's a metabolic liability for the cells. Well, why is that? Well, we've learned that they tend to overexpress a protein alternatively known as SRB1 or SCARB1. And this is a surface protein that takes in cholesterol through HDL particles. So again, coming back to the transcriptomic analysis, cholesterol biosynthetic enzymes are downregulated, just as we've seen for the gluconeogenic enzymes. But in contrast, SCARB1 is overexpressed. Now, these metabolic enzymes, these cholesterol biosynthetic enzymes are very consistently underexpressed, and that coincides with a decrease in steps in the pathway of cholesterol production, such as squalene. So the change in the expression of these enzymes coincides with the status of cholesterol synthesis. But this led us to revisit a genome-wide association study from some time ago that was looking for SNPs that predispose individuals to contracting renal cancer. And not too shockingly, 3 of them reside within EPAS1, which is the locus that encodes HIF2-alpha. HIF2-alpha, a well-recognized oncogenic driver of renal cancer. In fact, HIF2-alpha inhibitors are currently making their way through clinical trials, including at the Dana Farber. However, one of these SNPs resided in the locus that encodes SCARB1. And ChIP-seq experiments have indicated that the SNP, if anything, enhances HIF2-alpha recruitment to this locus, suggesting maybe HIF2-alpha overexpression leads to SCARB1 overexpression, and maybe the two are in some kind of epistatic relationship. So what does SCARB1 contribute to the disease? So again, we've looked at this in a lot of ways, and I'll just summarize with some simple flank tumors. Recall, we still are challenged with a good, usable, genetically engineered mouse model for renal cancer, but simply dialing up SCARB1 hairpins greatly attenuates tumor growth, and we've done this in many assays. This is just a flank tumor readout. This suggests another therapeutic opportunity. It turns out SCARB1 happens to be a co-receptor for hepatitis C virus receptors. And there are already well-validated inhibitors of SCARB1 as an antiviral approach. And sure enough, they can also attenuate renal cancer cell proliferation in vitro, and we've recently shown this in vivo as well. So unlike all of the other metabolic changes I mentioned, which lead to a significant downregulation of these various enzymes, making them hard to treat, SCARB1 is very consistently overexpressed, and we think contributes to this deposition of cholesterol and cholesterol esters, a hallmark of the disease. If we silence SCARB1, it causes these cells to very abruptly undergo cell cycle arrest. Now, because they have so much internal cholesterol, this leads to downregulation of the biosynthetic enzyme pathway, making the renal cancers complete cholesterol oxytrophs. And SCARB1 is essential for importing the cholesterol in the form of HDL, for example. So, as I said, we believe that even though the tumors consistently accumulate cholesterols and cholesterol esters, it is clearly a metabolic vulnerability or a liability, because to deal with it, as I mentioned, they're dependent on lipid droplet formation via perilipin, a lipid droplet coat protein, and a protein that enhances lipid droplet production. They're dependent on SCARB1 to import the cholesterols. The other thing they're dependent on is the conversion of the cholesterols to bioacids. And something that was recently appreciated is that renal cancer patients tend to excrete high levels of bile acids. And we believe that that's because they also overexpress very consistently one of the rate-limiting enzymes in bile acid synthesis known as HSD3B7. And it has prognostic information as shown here. And they tend to accumulate one of the steps in bile acid production, which I'll simply call 7-HOCA for short. And as I showed with SCARB1, HSD3 depletion impairs tumor growth. And we think it's because of this oxysterol buildup. So I see Marcia is there, which means I should stop talking soon. But, you know, just to quickly summarize, another consistent change is the overexpression of this bile enzyme, synthetic enzyme. Because it's present, this avoids a toxic cholesterol accumulation, in particular the product I mentioned, and its inhibition induces oxysterol accumulation and overt apoptosis. So, you know, right now everything sort of makes sense. They overexpress a cholesterol importer, they accumulate cholesterols, they park them in lipid droplets, they downregulate the biosynthetic enzymes as a compensatory response, and any excess gets converted to bile acids and excreted. But in my mind, it still begs the question, why do they so consistently overexpress SCARB1? And let me get my mouse to go here. We think that SCARB1 must be doing something else. We know it's connecting to AKT, PI3 kinase signaling, but we're still missing an important part of the puzzle. So we're now engaged in a CRISPR screen to see what other activity SCARB1 is engaging in that seems to make up for the fact that all this cholesterol ester accumulation really is a metabolic problem for the renal cancer cells, a consistent change, but one that they have to deal with at least 5 different ways that I can think of. And so that's what we're doing now. So just to briefly close, just wanted to mention members of the lab that did the study. So this is back when we could do fun things like have a lab party and a fun bar. We miss those days. But here's Pei Wei Wang, who did the work on FBP2 that I sort of glossed over. Here are Nico Scoli and Romain Riscal that are doing the cholesterol work that we're quite excited about. And Fuming Li is the first author of the paper we just published on liver cancer and stellate cells. So thanks again. I know people might be looking at this at home tonight in their pajamas as a recorded lecture, but feel free to email me with any and all questions and any suggestion. We're all ears. So thanks again very much. Maybe I could stop my screen share here. If I can. Yeah, here we go.
Thank you very much. So you have a number of questions.
Okay.
And you're right on time, Celeste. That was
good.
Really an awesome talk. And I'll read them and that way you can answer them.
Okay.
So, and I apologize in advance for mispronouncing any names. So Enthul Sobatar asks, do you think OPPP is stimulated in cancer cells in order to keep up with increased cell replication nucleotide synthesis?
I'm sorry, OPP?
That's what he's typing, OPPP.
OPP. I tell you what, why don't we put what you mean by that in the chat box and I'll go to another question. OPP. Marcia, do you know what he means by that?
I mean, maybe you can comment on how you think this regulation contributes to nucleotide synthesis or cell replication.
Which thing?
The HIF FBP regulation or
the HIF FBP1 interaction? Like
yeah.
How does this contribute to nucleotide synthesis? I think the most obvious thing I can point to is that if FBP1 is associated with HIF, there's decreased output of HIF, which includes things like glycolysis, which of course through the pentose phosphate pathway will contribute to nucleotide synthesis. I guess we've been thinking of it more as the proximal effects of glycolysis, but you're absolutely right. Anything that attenuates glycolysis would contribute to attenuation of all the building blocks for cell division. So, you know, again, to me, it makes great sense that there's this FBP-HIF interaction. If there's a challenging situation, this would conserve ATP, you know, it's sensing a buildup of AMP. It would conserve ATP. It would the buildup of AMP would release FBP, so you enhance glycolysis, you enhance the myriad of HIF outputs. Those glycolytic intermediates will feed into all the other pathways. But if FBP is present, then you have a more attenuated glycolysis. So again, it makes perfect sense why the tumors tend to lose FBP, because if you enhance glycolysis, you're going to enhance the building blocks that will indeed promote cell division. So if I understand the question, that makes sense to me.
Yeah. So the next question is the interaction between HIF-1 alpha and FBP1 limited to renal cancers?
So that's a very good question. We can see it in a number of cell types if we're under the right conditions. The way we think about it is that this would be relevant to the tumors that have an intact VHL system where oxygen is limiting. But it's important to contrast that, for example, with liver cancer or many soft tissue sarcomas where VHL is intact. And so we think that the FBPs, both of them, are probably talking to other nuclear factors. So the FBP-HIF interaction is really going to be dominant in either the setting of VHL loss or in the areas where HIF is beginning to accumulate. Now, I think all cells have a very little baseline, but where it really becomes quite robust is in the settings of either low O2 or VHL loss.
Okay, and so I'll just ask Steve Elledge's question because you touched on this already. Under what normal physiological conditions would HIF be activated in low AMP be used to modulate it?
So that's a good question, and not in a disease setting, but in a physiologic setting. So, a scenario that I could certainly think of would be, in exercise muscle, for example, where you could imagine a buildup of HIF, but then, bioenergetic challenges. So if there's a buildup of HIF that, FBP1 is so abundant, you might imagine they'd be associated to chromatin together. And then if the bioenergetic collapse further continues, FBP would be lost and HIF activity would be enhanced. Just what I can think of. I guess, well, I will say one thing. With FBP2, what we're really excited about, again, we're in the cancers, we're looking at proteins that shouldn't be there, right? So maybe a better way to answer Steve's question is to talk about the normal physiologic situation, such as muscle, where FBP2 is present, not FBP1. And we've been able to show that in muscle precursors, FBP2 is clearly in the nucleus, where the cells tend to be more glycolytic, but then clearly exits the nucleus as they further mature and form muscle fibers, where now they become very dependent on oxidative phosphorylation. And I guess I should say it's glycolysis versus oxphos. So the muscle system gives us the opportunity, and this is exactly what we're doing, to understand what regulates FBP2 shuttling in and out of the nucleus and how that contributes to the bioenergetic status. So like we have for FBP1, and we are programming that not only into tumors now, but into a number of normal tissue activities or other pathophysiologies. We're doing the same with an FBP2 allele.
So along those lines, I want to shift gears a little bit and ask the next question. Would you predict that FBPs, through their interaction with transcription factors, have a general role in senescence across other model systems or physiological systems?
So that's exactly what I think we're in a position to do. So I should point out that in the liver, where we exclusively deleted FBP1 in the hepatocytes, our system left FBP1 and other constituents of the liver. So it's still intact, for example, in the stellate cells. But in the liver, the effects on the stellate cells, activating them and then causing them to senesce, we can see without an oncogenic insult. We can see it without DEN. We can see it without a P53 change that causes cholangiocarcinoma. We can see it without the high-fat diet. So that's something FBP1 can do on its own. So our allele will certainly allow us to look at this in other tissues and investigate effects in other senescent scenarios. It's something we're very, we're so cancer-centric, but we would like to branch out there. It's a great point.
So Bruno Amati asks, does FBP2 dampen MYC activity at all, or a subset of MYC target loci? If the latter, what do these promoters have in common?
Great question. These are early days. The reason why we started to look at MYC is because of a transcriptome that, IPA, et cetera, analyses really highlighted a lot of MYC targets. So the transcriptome suggests it's broad, but I think we're only looking at this somewhat superficially. So those are exactly the things that we would like to do. So for example, an FBP2 ChIP-seq, Homer analysis, do e-boxes and other things come up? How much of the MYC output is affected? We think that both MYC repressed and MYC activated genes are being affected, but the thing that really jumped out at us is the output that has to do with mitochondrial biogenesis, mitochondrial function. And I should point out that observation where FBP2 is being added back in sarcoma is holding up in the liver where FBP1 is being lost. So the opposite results are reinforcing each other, if that makes sense. But these are the things we really do want to understand, is how broad are the effects on nuclear factors, where else in the genome are they sticking? And also just to be, we want to be really sure that this all, we're trying to be as careful as we can, to show that by every means available to us, that this is a highly conserved and important property of these proteins.
Thank you so much. So I think that I think that's a great place to stop. We have a number of other questions that we did not have time to get to, and I think there will be opportunity for Celeste to address those questions later in writing, so come back and check that. Thank you so much, and thank you everybody for participating today.
Appreciate it. And Marcia, just an operational thing, can I come back to this and look at the chats, or how do I do that?
I think there is opportunity for you to come back, and you could even type in your answers right now, and then they'll be recorded.
Oh, okay. Or what I could do is I could look at the chat and verbally answer, and people could check the recording. Does that work?
That does work.
Okay. All right. Thanks again. Nice to see you all.